Hi students. In the last lecture we discussed the journal pharmacology and the different routes of administration of the drugs and the journal principles of pharmacology. Today we are going to start with the pharmacokinetics. Kinetics, as we know, kinetics means movement. So pharmacokinetics is movement of drugs inside our body. When we take a drug or a tablet orally, what is going to happen to that drug? It is going to pass through our alimentary canal. It is going to be degraded by the HCl in the stomach. It is going through the intestine. It is going to get metabolized or broken down by the liver. That is pharmacokinetics by oral route of administration. What is going to happen if we are going to administer a drug via IV route? We are directly injecting our drug into the bloodstream, so the drug which is getting into the blood is 100%. So basically what is going to happen to the drug? That is pharmacokinetics. Now there are four main things which are going to happen once we are taking a drug. That is known as ADME, or absorption, distribution, metabolism, excretion. Now what is absorption? Absorption is when you are taking your drug. For example, if we are taking a tablet, it is going to disintegrate and it is going to be absorbed into the bloodstream or the systemic circulation. So when you are taking a drug and it is getting into the systemic circulation or the blood, that is absorption. Once absorption has taken place and the drug has reached the blood, what is going to happen? From the blood, the drug will be distributed into the cell, in the fluid outside the cell and in the fluid inside the cell. After distribution, it is going to get metabolized, and the primary site for metabolism is liver. Here it is going to be broken down into its metabolites. And once these are broken down into the metabolites, these metabolites help in the excretion of the drug. That is, the drug is eliminated from the body. So the first part of pharmacokinetics is absorption. Absorption, as we discussed, that when we take any drug, it gets released from its dosage form and it is entering into the systemic circulation or the blood circulation, so that it can be carried to different parts of the body and to the site of action. For this, when it finally gets into the blood and it has to be distributed to different parts of the body, it has to cross biological membrane. It has to cross a biological membrane. For example, this is a lipid bilayer. Suppose this is the extracellular fluid, and this is the intracellular fluid, and this is the cell membrane. This is an enlarged part of a cell membrane. If we talk about this in detail, it is about 100 angstrom thick and it is a bilayer of phospholipids and cholesterol molecules. If we take one part of this in detail, this is a phospholipid unit which is having a polar head and a non-polar hydrocarbon chain, and this polar head is hydrophilic in nature. That means it is water loving, whereas the non-polar tails, those are hydrophobic in nature, that is water hating, or lipophilic in nature, that is lipid loving. So this part will be water loving, whereas the internal part will be hydrophobic in nature. Now when drug present in the extracellular fluid has to cross this biological membrane and get inside the cell, how this process of this transfer from extracellular fluid to intracellular fluid will happen. So for this, there are certain specialized proteins in the biological membrane or the cell membrane. Those are known as intrinsic or extrinsic proteins. Intrinsic, as the name suggests, that they are lying within the whole of the cell membrane. They are embedded in the cell membrane, whereas extrinsic proteins are somewhere on the surface of the cell membrane. So because these are a tunnel like proteins, they can function as carrier proteins and transport channels, whereas these extrinsic proteins, because they are on the surface of the cell, they can function as receptors, antigens, recognition centers, etc. Now these proteins are involved in the transport of drugs across the membrane. There are two types of transport mechanisms. So there are two major types of transport mechanisms for the transport or absorption of the drug from extracellular fluid to intracellular fluid. These are passive diffusion and specialized transport. Passive diffusion is for lipophilic molecules, because our membrane is lipophilic in nature, so lipid soluble drugs can easily cross this membrane. And for this transport, for passive diffusion, there will be no utilization of energy or ATP molecules. The drug diffuses across the cell membrane in the direction of its electrochemical concentration gradient. That is, the movement of the drug will be from higher concentration to the lower concentration. Obviously, when we are giving a drug from outside and the drug which is present in the extracellular fluid will be more and the concentration will be more, and the concentration of the drug in the intracellular fluid will be less. So in the direction of the concentration gradient, the drug will passively move towards the intracellular fluid. Now there are certain factors which influence this passive diffusion of drug across the cell membrane. First is lipid water partitioning coefficient. The higher the partitioning coefficient, that is, the higher the lipophilicity of the drug, more will be the rate of transport. Second is concentration difference. Concentration of the drug that differs on the two sides. Higher this concentration difference on the two sides, faster will be the diffusion. Third is pH. pH, because most of the drugs are weak electrolytes and they are in two forms, ionic and non-ionic. As non-ionic forms are lipophilic in nature, so these are easily crossed across the cell membrane as compared to the ionic drugs. So the non-ionic drugs, because they are lipid soluble, they are readily absorbed inside the cell. And because of this phenomena, we can say that the weak acids are readily absorbed in the stomach. Why? Because in the stomach, because it is acidic pH, and these weak acids will not be ionized, so they remain in the non-ionic state, unionized state, and they are readily absorbed. Whereas weak bases are readily absorbed from the intestine, because the intestine, again because of the alkaline pH, they will not get ionized, and non-ionic form will easily get absorbed from the membrane. Now, the second transport mechanism through which drug absorption takes place other than passive diffusion is specialized transport. The major specialized transport is carrier mediated transport. There are other, but they like phagocytosis or pinocytosis, but they contribute little. What happens in phagocytosis is when a drug molecule comes and attaches to the membrane, and this membrane, it engulfs this drug molecule, goes inside and leaves this drug molecule inside the cell. But the major carrier transport is the specialized transport, in which there is a carrier protein on the cell membrane, and when a substrate or the drug molecule comes and attaches to this carrier protein undergoes a conformatory change and transports or transfers this drug molecule towards the intercellular fluid of the cell. There are two types of carrier mediated transports. One is facilitated transport and other is active transport. As we discussed, there are two major types of carrier mediated specialized transport. Those are the facilitated diffusion and active transport. First, coming to the facilitated diffusion. In facilitated diffusion, just as passive diffusion, the substrate or the drug molecules will be passively carried, that is, without the use of energy. They are carried from higher concentration towards lower concentration, that is, in the direction of electrochemical gradient. But what is the difference between facilitated and passive diffusion? That is, the transporters which are involved in facilitated diffusion, they belong to super family of solute carrier transporters. We call it as SLC. That is, these transporters are specific for a specific solute. They will carry only a specific substrate. For example, GLUT4. These are the transporters for glucose molecules. Only the glucose transporters, these are the glucose transporters which facilitates the entry of glucose into muscle and fat cells. They will just attach to a glucose molecule and they will transfer this from higher to lower concentration. This is known as facilitated diffusion, in which the transporters bind to a specific substrate. The next type of specialized carrier mediated transport is active transport. Active transport means because it is utilizing energy, it is utilizing ATP molecules to transport the substrate or the drug molecules. and this transport is against the concentration gradient, that is from low concentration to high concentration. So it is working actively by using energy or ATP molecules, and these are transporting the substrate molecules from lower concentration gradient to high concentration gradient. Active transport is of two types. First is the primary active transport, and second is the secondary active transport. What is happening in the primary active transport? The energy for the transport of the molecules is derived from ATP molecules. ATP is hydrolyzed to ADP. Adenosine triphosphate is converted to adenosine diphosphate, and a substrate molecule is carried from low concentration towards the higher concentration. This is known as primary active transport. Now, what is happening in case of secondary active transport? Here, ATP molecules are not used to transport the substrate molecules. So what will be used? It is the movement of another solute. That is, the energy to pump one solute is derived from movement of another solute, which is mostly it is sodium ion. That is, the movement of your drug or your substrate will be done from the energy which is obtained from movement of another ion or solute. These are of two types. Suppose this is your carrier and it is taking your sodium ion and your drug molecule in the same direction, then this is known as symport. And if your carrier is taking your sodium ion towards one direction and drug molecule to the opposite direction, then this is known as antiport. Now we are going to discuss about various factors which affect the absorption of drug into the systemic circulation. First among them is the aqueous solubility. Now, in order to get absorbed into the bloodstream, it is very important for the drug to dissolve in an aqueous biophase if the drug is in solid dosage form. If you are taking a tablet, it is important for that tablet to get released. That is, the active ingredient should be released after the tablet is getting disintegrated. It should get dissolved into the aqueous biophase and then it will be absorbed into the bloodstream. Therefore, the increase in the aqueous solubility will increase the rate of absorption. The second factor which affects the absorption of drug is concentration. Now, we studied that passive diffusion is taking place because of the concentration gradient. We do not require any energy for passive diffusion. We just need that there should be a concentration gradient, concentration difference across the two membranes. And if this concentration gradient or the difference between the two membranes with respect to concentration of the drug is higher will be the rate of absorption. Similarly, for area of absorbing surface, for example, if you apply a moisturizer or any ointment on one part of your finger and you are not rubbing it around the other parts, then it will take a lot of time and to get absorbed into the skin. However, if you will apply your moisturizer or your ointment to a larger part of the skin, then it will be absorbed much faster. Therefore, the area, if you increase the area of absorbing surface, it will increase the rate of absorption. Now, vascularity. If that area where you are administering your drug is highly or richly supplied with the blood vessels, then there are high chances that your drug will be absorbed faster into the bloodstream. The last factor among the influencing factors is the route of administration. Now, the different routes of administration affect the absorption differently. The most of the time, your oral route of administration takes the longest to to get the drug to be absorbed into the bloodstream, because your drug has to face different barriers such as your acidic pH in your stomach and your alkaline pH in your intestine. Then it has to cross that first pass metabolism, and then it gets into the blood. So different routes of administration will affect the rate of absorption differently. As we have discussed so much about absorption, that absorption is when we are administering a drug, it gets absorbed from its site of administration to the bloodstream. But have we thought that how much amount of drug is being passed into the bloodstream? For example, if we are giving 100 mg of drug, how much amount of this 100 mg is being transported into the blood? Whether this 100 mg is completely transported or how much amount is being transported. This is known as bioavailability, which is denoted with F. Bioavailability is the amount of drug. It is the amount of drug that reaches the systemic circulation in unchanged form. And for IV route, because we are directly injecting the drug into the bloodstream, this bioavailability is 100%. But for other routes of administration, for example, oral route of administration, it cannot be 100%, because it is possible that drug is not completely absorbed, or it is undergoing first pass metabolism. And if you are giving a subcutaneous or intramuscular injection, it is possible that local binding of the drug is taking place and complete concentration, a whole of the drug is not getting absorbed into the systemic circulation. So how you can calculate systemic bioavailability? There is a formula, that is amount of drug in systemic circulation divided by the dose you have administered. The total dose you administered and the amount that reached into the blood circulation. If you divide this, you will get your bioavailability, that how much amount of the drug has reached into the systemic circulation out of the total dose administered. Now there is another important term, that is bioequivalence. Now what is bioequivalence? For example, there are two manufacturers and they are producing same drug, or the same manufacturer is producing the same drug in different batches. Now we have to compare the bioavailability of the two drugs. For example, there is drug 1 which is giving you bioavailability, and it is reaching this level of this concentration in the blood. And another drug from a different manufacturer, a different lot, is reaching different level in the blood. Maybe the drug they are having same amount of the drug, that is they are chemically equivalent, but because they are yielding different blood levels, they are biologically inequivalent. So these two drugs are they are considered non-equivalent. These are not bioequivalent. So two preparations of a drug are considered to be bioequivalent when the rate and extent of bioavailability of the active ingredient from them is significantly different. Now this result may be because of the different conditions which were given to the drug, or the manufacturing processes of the two different companies were different. For example, they are using the different force in compressing the tablet, because of which there is a delay in the disintegration if the force is more. So this the bioequivalence will vary. In this lecture we discussed about the pharmacokinetics and took up bioavailability in detail. In the next lectures we will be taking up distribution, metabolism and excretion in detail. So that's it for today. Thank you.